Hello dear students, welcome to Nanomongo online classes. In the last class, in a chapter molecular basis of inheritance, we discussed translation, the process of polymerization of amino acids based on the sequence of nucleotide sequence present on mRNA. Okay, so that the concept we have discussed. What are the requirements as actually used in a translation process? The translations are 4 steps: activation of amino acid, initiation, elongation, next termination. What are the factors that are involved in such steps? We also discussed them in the last video class. Today we will discuss the next topic, that is expression of gene. In a previous video I have discussed that gene actually it is an information present in segment of DNA. Okay, remember the gene can be expressed in number of forms. Or levels, because gene is a particular information. It could be expressed in transcriptional level. Transcription level means DNA is in the mRNA, and then the particular information copied, expressed. Processing level, in case of eukaryotes, this is splicing, where this mRNA gets modified. Actually here we can say introns where ring. Okay, so modification or processing level where nadu iruvanta non-coding sequences again. Next, during transformation from nucleoplasm to cytoplasm, ಅಲ್ಲಿ ಕೂಡ ಏನಾಗ್ತೈತಿ ಇದು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗ್ತೈತಿ, and the lastly it expressed in a translation. So in translation it expressed in a amino acid sequence. This is the final expression. So the final expression is a formation of polypeptide. So the information which is present in DNA, ಯಾವ information DNA ಇರ್ತೈತಿ, ಯಾವುದು amino acid chained or polypeptide, so ಇದೆಲ್ಲ processing levels, ಇದೆಲ್ಲ levels, expression levels ಮುಖಾಂತರ ಲಾಸ್ಟ್ ಗೆ ಏನಾಗುತ್ತೆ amino acid sequences or particularly polypeptides ಇವೇನಾಗ್ತವೆ ಫಾರ್ಮ್ ಆಗ್ತವೆ. So expression of gene, expression of gene, it will not take place whenever there is not necessary. Necessary irlada time the genes express agadi. Remember, the ultimate product of gene expression is ultimate product. Gene expression the ultimate product ಏನೈತ್ರಿ, one particular protein. So whenever there is a necessity, gene will express, it produces that protein. Whenever there is not necessary, this gene will not express and the protein will not form. You can take the example in a bacterial culture. In a bacterial culture, if you have added lactose, the bacteria culture of lactose you add madidanga, lactose is converted by bacteria into glucose and galactose. This glucose is used in the form of energy. Energy produced by glucose ka use. That means lactose is also a source of energy. So whenever you will add the lactose, bacteria utilize this lactose. For this breakdown ಆಗಬೇಕಂದ್ರೆ ಕಂಪಲ್ಸರಿ ಏನು ಬರಬೇಕ್ರಿ enzymes ಬರಬೇಕು. Remember, enzymes are also polypeptides. Enzymes ಕೂಡ ಏನದಾವೆ polypeptides. So that means they are contains a particular information. That information is in a particular genes. So this gene has to be expressed. Whenever bacteria got the lactose, the genes which are responsible for the production of enzymes and the enzymes which break down the lactose into glucose and galactose, these enzymes has to express if in a medium there is no lactose, there is no source of energy, then there is no need of production of these enzymes. That means there is no need of expression of these genes. Okay, so what happens in that? Whenever there is a necessity of expression of genes, the genes are expressed in a particular polypeptide or protein. If not, they will not express in a particular polypeptide. So this concept was given by Francis Jacob and Jacques Monod. So this concept is called Jacob and Monod concept. Okay, so this concept they have given on E. coli bacteria by introducing lactose. Okay, so to break down the lactose, E. coli bacteria produces three types of enzymes: beta galactosidase, trans-acetylase, and permease. These three genes, they are produced from these three enzymes actually. These three enzymes, they are produced from same genes. That means this gene has the information of these three enzymes. So that on that note, this gene is considered as polycystonic gene because one gene gives three products. Okay, so polycystonic genes. So beta galactosidase, they divide lactose into glucose and galactose. Permease, they will take more lactose inside the cell. More lactose gap inside the cell. This beta galactosidase convert that lactose into glucose and galactose. ಎಷ್ಟು ಲ್ಯಾಕ್ಟೋಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಒಳಗೆ ಬರ್ತೈತಿ ಅಷ್ಟು ನಿಮ್ಗೆ ಏನು ಸಿಗ್ತದೆ ಗ್ಲೂಕೋಸ್ ಮತ್ತು ಗ್ಯಾಲಕ್ಟೋಸ್ ಮಾಲಿಕ್ಯೂಲ್ ಸಿಗ್ತವೆ. ಸೊ ಪರ್ಮಿಯೇಸ್ ಏನು ಮಾಡ್ತೈತಿ ಲ್ಯಾಕ್ಟೋಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಒಳಗೆ ಬರಾಕ ಹೆಲ್ಪ್ ಮಾಡ್ತೈತಿ. ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸೈಡೇಸ್ ಲ್ಯಾಕ್ಟೋಸ್ ಗ್ಲೂಕೋಸ್ ಮತ್ತು ಗ್ಯಾಲಕ್ಟೋಸ್ ಒಳಗೆ ಬ್ರೇಕ್ ಮಾಡಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತೈತಿ. ಟ್ರಾನ್ಸ್ಅಸಿಟೈಲೇಸ್ ಯೂಸ್ ದ ಫೀಡ್ಬ್ಯಾಕ್. ಟ್ರಾನ್ಸ್ಅಸಿಟೈಲೇಸ್ ಏನು ಮಾಡ್ತಾವ್ರಿ ಫೀಡ್ಬ್ಯಾಕ್ ಮೆಥಡ್ ಕೊಡ್ತಾರೆ. ವೆನ್ the requirement is full, there is no need of such expressions, so they need to stop that process. Other purpose ಏನಾಗ್ತೈತಿ feedback transacetylase help ಮಾಡಿ. Next, the eukaryote cell, this E. coli bacteria is a prokaryote. E. coli bacteria ಏನೈತಿ prokaryote. And remember, for expression of genes ಇದಾಗಬೇಕಂದ್ರೆ ಕಂಪಲ್ಸರಿ ಏನಾಗಬೇಕ್ರಿ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಬೇಕು. ಕಂಪಲ್ಸರಿ ಏನಾಗಬೇಕ್ರಿ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಬೇಕು. ಸೊ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಬೇಕಂದ್ರ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನಲ್ ಯೂನಿಟ್ ಒಳಗ promoter region ಮೇಲೆ RNA polymerase ಅಟ್ಯಾಚ್ ಆಗಬೇಕು. ಸೊ RNA polymerase ಪ್ರಮೋಟರ್ ಮೇಲೆ ಅಟ್ಯಾಚ್ ಆಗಬೇಕು. ಆಗಬೇಕಿಲ್ಲ ಮೀನ್ಸ್ necessity to RNA polymerase attaches on promoter region. If there is no necessity of expression of genes, RNA polymerase will not attach on promoter. So the attachment of RNA polymerase to this promoter region is controlled by a particular different set of genes. Those genes produces proteins. These proteins are called regulatory proteins because the set of genes are regulatory genes plus operator genes. So the activity of one gene is controlled by a set of other genes. So these other set of genes, they are referred as operons. You can know operons. So this operon controls the expression of structural gene. Whether to synthesis mRNA sense mRNA produce ಆಯ್ತಂದ್ರ ಮುಂದೆ ಏನಾಗ್ತೈತಿ ಪ್ರೋಟೀನ್ ಆಗೇ ಆಗ್ತೈತಿ. So whether to express this structural gene in the form of their products or not. On that note, these regulatory proteins, they act as activators. Activator means to initiate when the necessity is there, or they act as repressors to terminate means there is no necessity. If there is no necessity, no lactose is there, regulatory protein act as a repressor. The mechanism expression of this gene which produces this product, they terminate. They stop. How they will stop? They will not let this RNA polymerase to attach a promoter. Actually, the promoter is not attached by RNA polymerase. It is attached on operator region. So these are the sequences which present besides the promoter. Okay. So if RNA polymerase attached on promoter region, then the transcription starts. If these regulatory protein will not let the RNA polymerase to attach on promoter, regulatory proteins RNA polymerase operator region attach ಆಗ ಕೊಡ್ಲಿಲ್ಲ ಅಂದ್ರೆ regulatory proteins repressor ಜೊತೆ ವರ್ಕ್ ಮಾಡ್ತಾವ್ರಿ. they terminate. ಇಲ್ಲಿ gene express ಆಗಂಗಿಲ್ಲ, product ತಯಾರು ಆಗಂಗಿಲ್ಲ. if regulatory proteins let the attachment of RNA polymerase to operator, regulatory proteins RNA polymerase operator region attach ಆಗಕ್ಕೆ ಕೊಟ್ರ ಅವು activators ಆಗ್ತಾವ, initiate ಮಾಡ್ತಾವ, transcription ಆಗ್ತೈತಿ, genetic information copy ಆಗ್ತೈತಿ, ಮುಂದೆ translation ಒಳಗೆ product form. So letting the RNA polymerase to the operator region, RNA polymerase operator region attach ಮಾಡಬೇಕಂದ್ರ different mechanism ಆಗ್ತದೆ. So that particular set is given in an operon concept. Operon concept. So what happens in operon? It is a set of genes. Operon ಏನೈತಿ? ಒಂದು set of genes ಐತಿ that controls the expression of other genes. In operon, I genes are present. ಏನಂತೀವಿ ಇದಕ್ಕೆ? Regulatory genes ಅಂತೀವಿ. I ಅಂದ್ರೆ inducer or inhibitor ಅಂತ confuse ಮಾಡಬೇಡಿ. Regulatory genes. They always produce regulator proteins. ಮೊದಲೇ ಹೇಳಿದ್ರಿ regulator protein activator ಆಗ್ತಾವ್ರಿ, repressor ಆಗ್ತಾವ. Next, operator. Operator genes. The genes which are present besides the promoter region and is always attached by regulatory proteins. They are always attached by regulatory proteins. Remember, if regulatory protein attaches the operator region, no RNA polymerase attaches to the operator region. Okay. Then next, structural gene. So this structural gene, it contains Y gene, A gene, Z gene. Y gene produces beta galactosidase, A gene produces permease, Z gene produces transacetylase. Polycystonic condition. Remember, from here to here, Y gene from here to here information, A gene from here to here, Z genes. Together they are structural genes. Okay, so this is the concept of operon, which was proposed by Jacob and Monod. This operon has two concepts. Operon, when it is switched on, means activator condition. When in repressor condition, operon will be switched off. This concept was given by Jacob and Monod in E. coli bacteria by adding the lactose medium. Okay, how the operon get switched off and operon get switched on? What are the these factors that helps the attachment of RNA polymerase to the operator region even in the presence of regulatory proteins? Because regulatory proteins, they have strong affinity towards the operator region. Okay, so what are the other conditions in operon, lac operon concept or lac operon model? Switch off and switch on mechanism we will discuss in the next class. Okay, thank you.